So we're going to be continuing our endocrine discussion, and today we're going to focus a little bit more on homeostasis. We're still going to review the pathway some. So remember, the pathway starts with our reception, okay, where the ligand, that signal molecule, remember it's also called a ligand, and in this case we're going to focus on hormones. So that ligand bonds to the receptor, which will start the transduction. And remember, the transduction is where that signal gets amplified and gets changed into an actual cellular response. And that response can be a variety of things. So let's start first with the receptors, just as a few reminders of some of the receptors that we've looked at. Okay, so remember we talked about a G protein. Okay, when that G protein gets activated, it will start the second messenger, the tyrosine kinase, which will phosphorylate, and then it will activate multiple pathways. Okay, as well as the ion channel receptor, which will open and close in correspondence with the binding from the ligand. And then remember, an intracellular receptor, if it was a steroid-based hormone, where they could fit through that cell membrane and they would bind to something within the cell, that would then trigger the cellular response. So just some more reminders of these receptors. So again, in case you forgot, here's your G protein. Remember, and that got activated when the ligand binds to it, and that activates the G protein, which is turns on that GTP molecule, okay, and then that will activate the enzyme that will trigger the response of the cell, okay, as well as the over here we have the ion channel. Okay, so this ion channel, here's my ligand coming in. When it binds to it, my ion channel opens. When it releases, my ion channel closes. And then here's that intracellular receptor inside the cell there. The steroid-based hormones cross over and they go and it affects gene transcription directly. Okay, and so your reception, okay, is when the signal is coming in and it's binding to it. It's what's triggering that transduction pathway. And so then that transduction pathway is going to be the conversion of that signal. So here's our transduction pathway here. Remember that the job of that transduction pathway is to either amplify the signal, okay, to transfer it, okay, but when we're transducing something, we're turning it into something else. So let's take a look at a short video clip that'll really show the importance of that amplification.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activates several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
So hopefully you saw there in the video how the amplification process is so important, how it's going to help encourage that cellular response quickly and at a larger scale. So we have three basic types of responses that can happen due to that the signal reception and the transduction pathway. Okay, we can get activation of an enzyme. So like you saw in the video, you can have production of the enzyme, things that are currently in the cell, and just get them turned on and making the enzyme. You could have gene activation. Things can act as transcription factors. Okay, wow. Okay, so things could act as transcription factors. And if y'all remember from your freshman year, transcription was the first part of protein synthesis, right? And we take that DNA and turn it into a messenger RNA. So we can have things acting as transcription factors, as well as changes in the cytoskeleton and rearrangement of the cytoskeleton. These are kind of our three basic responses. Everything will be kind of a version of these. So we have two basic kinds of proteins, or hormones, I'm sorry. So we're going to be focused on the hormone for just a minute here, the actual signal, the ligand that's coming in there. We've got water-soluble and water-insoluble hormones. And so remember, water-soluble, they would be able to dissolve in water. Water-insoluble would be unable to dissolve in water. So our water-soluble hormones are going to be our protein-based hormones, amino acid derivatives, polypeptides. Some examples of this would be epinephrine or adrenaline. Okay. Insulin would be another example of a protein-based or water-soluble hormone. Our water-insoluble hormones, so these are not going to dissolve in water, so these are going to be lipid-based. These are going to be our steroid hormones. Okay, so examples of these would be androgens, testosterone, thyroid hormone, so thyroid hormone, testosterone, the hormone version of vitamin D. Those are all water-insoluble or steroid hormones. And so they're going to work a little bit differently. So our water-soluble proteins, and these are our protein-based hormones, these are going to travel in the blood, and they're going to be able to travel freely in the blood. They're not going to require any carriers or any helps. Where our lipid-soluble hormones, okay, our water-insoluble hormones, those are going to require a transport protein as they travel through the bloodstream. Remember, hormone signaling is a form of long-distance communication, so they have to travel in the blood. So these water-soluble ones travel freely in the blood. Water-insoluble have to travel bound to a transport protein. And then when they get to their cell, when they get to their target cell, they will have a little bit of a different effect. Okay, or not necessarily a different effect, the way they're going to be received by the target cell. So your water-soluble or your protein-based, they cannot cross that cell membrane. Remember your cell membrane made up of all those phospholipids with those hydrophobic tails. So they're not going to let a protein that has a charge, remember it will probably have some form of charge and it will probably be too large. They're not going to let that go through the cell membrane. So your water-soluble hormones, they're going to have to bind to surface receptors. Okay? And so when they bind to surface receptors, they'll trigger their response. They can either do a second messenger system, or they could possibly come in and do gene regulation. Okay? Your water-insoluble or your steroid-based hormones, once they've traveled to the cell via the transport protein, they usually are nonpolar and they're smaller, so they're able to fit through this cell membrane. Okay, so they'll squeeze in through that cell membrane, and they will use remember intra is within, they will use an intracellular receptor. Okay, so their receptor, instead of being on the cell surface, will be inside of the cell in either the cytoplasm or possibly all the way into the nucleus. And then that hormone protein complex, remember, acts as a transcription factor. And so that triggers gene regulation in that cytoplasmic response. So let's look at a couple of examples. So this example here is epinephrine. Epinephrine is a water-soluble or a protein-based hormone. So you can see it binding here to its surface receptor. It uses a G protein receptor. So it binds to the surface receptor on this target cell. And then that triggers our enzyme, triggers our pathway, Cascade. Okay, so when it triggers that pathway Cascade, it amplifies the response, so we get a significant response. And then we'll have our cellular response based on that, okay, based on the binding to the cell surface receptor. So looking at our steroid hormone or our water-insoluble hormone example, the response here, again, like I said, is usually gene expression. So in this case, we have a steroid hormone coming in. Let's say let's talk about mustache. Let's talk about facial hair in males as they go through puberty. Remember, our testosterone, estrogen, they're responsible for our secondary sex characteristics. So if this was testosterone here, okay, headed into the cell, remember, not all cells are going to have receptors for testosterone, but in males, there are skin cells in their face that have receptors for testosterone. And so the testosterone is able to cross the cell membrane. Okay, it binds to its receptor. That hormone receptor complex acts as a transcription factor. So it's going to turn on some genes. And in this case, it'll turn on genes that make the protein keratin. Keratin is the hair that's going to be found in the mustache. And so it'll make the protein keratin, and they'll start producing the hair due to that signal from the steroid hormone. And so these responses usually take a little bit longer than a protein-based hormone because they have to trigger gene expression and the protein has to be made. It's not necessarily turning on something that's already present in the cell and just turning it on or turning it off. So I'd like you to pause for just a minute and try to complete this chart. It's just a simple summary comparison chart to protein versus steroid hormones, but go through and make sure you get this chart into your notebook and kind of summarize everything for you there. So in talking about these hormones, we need to talk a little bit about their response that they cause in the cells. So hormones can affect different cells in different ways. They can affect one type of cell and do opposite effects. And the hormone effects are going to be dependent upon the receptors that are present on the cell surface. Okay, so those will determine the hormone's effect. Okay, so the receptors that are present in the cell surface, as well as the transduction pathway. The transduction pathway inside of the cell may be different. It depends on what is available. And so if the transduction pathway is different, the cellular response will be different. For instance, if you look here on the left, okay, the protein kinase A, the second messenger response there, can either inhibit glycogen synthesis. It'll inhibit the glycogen synthesis as well as promote the breakdown of glycogen. So it's causing two different responses at the same time. Looking at the bottom right here, same thing. We've got epinephrine again. Okay, so using our epinephrine response here. Epinephrine is released in response to fight or flight. When you're getting ready, you're really embarrassed, you're afraid, you're scared, you're stressed out, you release epinephrine or adrenaline. And adrenaline affects your body in all different ways. Okay? And so this was one of the ways it could affect it here. Okay? It encourages the breakdown of glycogen to release glucose, to raise blood sugar so your cells have enough nutrients to be able to do cellular respiration. It also slows the formation of glycogen so you're not pulling glucose out of the bloodstream. If we look down here, okay, we've got different receptors. Okay, so in this case, we've got blood vessels, which both contain smooth muscle, but in skeletal muscle, they have beta receptors, and in the blood vessels leading to the skeletal muscle, there are beta receptors, and in the blood vessels leading to the intestine, there are what are called alpha receptors. So the beta receptor actually triggers the vessel to dilate, while the alpha receptor triggers the vessel to constrict. So it's the same hormone, it's the same signal, the same ligand, okay, but because the receptors are different, the response is different. So let's take a look at a couple of particular protein pathways. Okay, so we'll look at responses that they can cause, and we'll look at a negative versus a positive feedback loop. Okay, so your basic hormone path is that it's secreted from a cell. Okay, so it's secreted from whatever gland is secreting that particular hormone. That hormone binds to the target cell based upon the receptor that is there. Okay, and so when it interacts with that receptor, it causes our physiological response. We have we trigger our transduction pathway and trigger the response. Let's take a look at what's called secretin. Okay, so secretin is found in the duodenum, which is part of our digestive tract. So if we're looking at this here, so what our stimulus, okay, is that there is a low pH in the duodenum. So when the stomach begins digestion, it releases all those acids. Remember, acids are low on our pH scale. And so when the contents of our stomach enter our intestines, they're acidic because they came straight from the stomach. And so that stimulus, that lowering of the pH, triggers the duodenum, okay, to release the hormone secretin. Okay, and so secretin then will enter the bloodstream and it will travel to the target cell. And the target cell in this case is the pancreas. So it travels to the target cell. Okay, so the pancreas at the target cell, and it will elicit a response from the pancreas. And the response is that the pancreas is going to release bicarbonate. Okay, and bicarbonate is alkaline. And remember, alkaline means that something is more basic. So bicarbonate is alkaline, and so that pancreas secretes the bicarbonate into the duodenum, which will help raise the pH. And when that pH gets raised, so the bicarbonate's released, okay, so now my pH goes up, and so this is negated. Okay? I no longer have a low pH in the duodenum. And so if there's no longer a low pH in the duodenum, then there's no need to release the secretin. And so this is an example of what's called a negative feedback loop. Okay? So let's look at the difference between a negative versus a positive feedback loop. Okay, so a negative versus a positive feedback loop. In a negative feedback loop, it inhibits the response by reducing the initial stimulus. So go back to your secretin example. So the initial stimulus in secretin was that I had a decrease in pH. Let's put all of this in the same color. So I had a decrease in pH. That was my initial stimulus. Well, when I released the secretin, and that caused the pancreas to release the bicarbonate, now my pH goes back up. So I am reducing the initial stimulus. Okay? I'm changing that initial stimulus. My pH went low, I'm bringing it back up. If my pH went too high, I'd bring it back down. So the negative feedback loop is going to inhibit a response because it changes that initial stimulus. By raising my pH, I no longer need to release the secretin. So I inhibit my response because I have reduced my initial stimulus. A positive feedback loop reinforces the initial stimulus to produce an even greater response. For instance, if my pH was low, okay, then that pH, I'm going to reinforce my initial stimulus of my low pH, okay, and I'm going to encourage my pH to go even lower. That would be a positive feedback loop. And we don't have a ton of examples of that in our body. In our body, examples would be blood clotting and uterine contractions. We'll look at a picture for uterine contractions in just a second. Another one that would apply to all mammals would be oxytocin. Okay. Oxytocin is the hormone that's responsible for milk letdown in mammals. And so the suckling response from the offspring of the mammal encourages more milk to be let down. So the oxytocin releases the milk. Okay. The suckling then causes the release of more oxytocin, so you release even more milk. So if you look here, this is an example of a positive feedback loop, again with oxytocin. So in this example here, you've got oxytocin, okay, that stimulates the uterus to contract. So oxytocin there stimulating the uterus to contract. Okay, and so this becomes a positive feedback loop that encourages even more contractions. Okay, that release of oxytocin in the uterine contractions signals the release of more oxytocin so that labor will progress and it will continue, and those contractions actually get progressively stronger. So it's reinforcing the stimulus of the uterine contraction, where a negative feedback loop would turn that stimulus off. Okay, it would inhibit the response by reducing the stimulus. So we're going to focus a lot on negative feedback loops because they are more common. So as the example we're going to be looking at right now. So as you may have noticed, many hormones work in pairs to help maintain homeostasis. So this right here would be homeostasis, normal blood glucose levels. Okay, and so right here we're going to be focusing on insulin and glucagon. Okay, and so insulin is going to help lower blood sugar. Okay, so insulin decreases blood sugar, whereas glucagon is going to help raise blood sugar. So these two are going to work together. I can't spell homeostasis. These two are going to work together to help maintain homeostasis. So let's look at a couple of stimulus examples. For instance here, okay, if my blood glucose rises, I've eaten something. I've eaten a piece of candy. I've eaten a pasta dinner. I've eaten a big meal in general, whatever. Okay, if my blood glucose rises, that's going to be my stimulus, is my increase in blood glucose. So there's my stimulus. That stimulus, okay, is going to cause a response in the form of the insulin release. Okay, insulin is one of those protein-based hormones. So insulin is going to bind to receptors on the body cells. It'll bind to receptors on the liver, and that will trigger them to pull the glucose out of the bloodstream. So they'll pull the glucose out of the bloodstream, and the liver will store it as glycogen. The cells, depending upon the kind of cell, some may store it as glycogen, some may store it as fat. and that's going to lower my blood glucose level. Okay, so I have inhibited my response because I've reduced my initial stimulus. My blood glucose level was too high. Okay, I lower my blood glucose level, so that reduces my initial stimulus, and so that's going to actually turn it off. I'm no longer going to secrete insulin. So that would be a negative feedback loop. Okay, let's look at the stimulus down here of my blood glucose level falling too low. So in this instance, my blood glucose has fallen too low. That's my stimulus. So now my alpha cells of the pancreas are releasing glucagon into the blood. And so that's going to go to those liver cells and those body cells and tell them to release that glucose they stored. So break down the glycogen that's been stored there and help raise blood glucose levels. So again, I'm reducing my stimulus. It was too low, I'm raising it back up. And that's going to turn off my release of glucagon. So again, another negative feedback loop. So let's take a look at then what happens when this gets disrupted. So diabetes mellitus occurs when this insulin pathway is going to be disrupted. Essentially, diabetes mellitus is a insulin deficiency or a lack of response to insulin. Okay, so you either have you're not producing enough insulin, or your target cells are no longer responding to the insulin very well. Okay, so it can be a decreased response to insulin as well. And so insulin is divided, There are two kinds of diabetes mellitus. Okay, we've got type 1 versus type 2. Okay, so type 1 would be here with my insulin deficiency. Type 1 is an autoimmune disorder, so your body has attacked your beta cells of the pancreas. So the beta cells are destroyed in insulin dependent. They're called insulin dependent type 1 diabetics. So they don't produce any insulin. Type 2 diabetes is usually a decrease in response. So either there's not enough insulin being produced, or we've got a decrease in response to the target cell because the insulin receptors have been altered. Essentially, they become desensitized to insulin. And so what this does is it's going to, if these insulin receptors, if you look at this, hopefully you can tell me what kind of receptor this is. Okay, hopefully you realize it's a tyrosine kinase receptor. So these insulin receptors essentially are not responding to the insulin in type 2 diabetes, or in type 1 diabetes, there's no insulin being produced at all. And so our blood glucose levels with diabetes mellitus, our blood glucose levels stay high. And this is a problem because the sugar, the extra sugar in the blood, will coat the red blood cells. So when it coats the red blood cells, it makes them stiff. And so if they're stiff, that interferes with our circulation, which can cause cholesterol and plaque to build up. People that have diabetes usually, as it progresses, they develop circulatory problems. Okay, so we need to be able to apply how the normal pathway should work, and then if there is some kind of deficiency or interruption to that pathway. So in your example, you also need to be thinking of how, if there was too much or too little of this particular hormone, what could it do to our bodies?